everyone, my name's Izzy Lake. I'm a graduate student from Newcastle University in the UK. The next two presentations you're going to watch is part of the Sea Turtle Research and Conservation Programme, which has been led by Annabelle Brooks for the last 5 years. My master's project is on the anthropogenic impact of seagrass and foraging habitats of green sea turtles.
Hi guys, I'm Logan Zynet. I'm the Reef Ecology and Restoration Technician at CEI, and I'm proud to introduce the coastal team for spring 2017.
Good morning, everyone. My name is Alex Weber.
I'm Addie Brown.
I'm Annie McGill.
I'm Will Unterline.
I'm Olive Cowan.
I'm Claire Cordova.
And I'm Sawyer Goldman.
And throughout the duration of our research study, we have been aimed in focusing on the anthropogenic impact on the growth rate and biomass of Thalassia testudinum, or turtle grass, in southern Eleuthera. Coastal ecosystems around the world are some of the most biologically diverse and unique ecosystems you'll find. Thousands of species rely on their shallow waters and sunlit conditions to flourish, and the foundation of these ecosystems are the primary producers. Primary producers are autotrophs that utilize photosynthesis to garner energy from the sun. And one of the most commonly found marine autotrophs is seagrass. Seagrass is responsible for 11% of the organic carbon buried in the ocean, and on an annual basis, 1 acre of seagrass alone can sequester 7,401 pounds of carbon. And to put that into perspective, that's enough carbon, that's equivalent to the amount of carbon emissions your car would give off on a road trip across the United States and halfway back. So this figure here represents the global distribution of seagrass and its diversity. As you can see, the darker areas are places with higher diversity of seagrass. And you can see that seagrass is found in every single ocean and borders nearly every coastline, which is significant because our research can be applied on a global scale. So seagrass is this photosynthesizing autotroph that is high in protein and very fibrous, making it an ideal diet for mammals, crustaceans, mollusks, and invertebrates. It comprises nearly 93% of the green sea turtle's diet, and their digestive efficiencies are enhanced due to the lower lignin levels in the seagrass that has been regrazed. And threats to seagrass include sewage runoff, fertilizers, and erosion.
As Alex mentioned, seagrass is a global organism and therefore provides many ecosystem services. One of those ecosystem services is storm protection. So the parts of the seagrass are the blade, the sheath, the rhizome, and the root. The rhizome holds together the sand and prevents any sedimentation that could occur during natural disasters. Seagrass also acts as a nursery for any juvenile organisms, such as juvenile stingrays, juvenile turtles, juvenile fish, or any other organisms that could find a food source or protection from predators in seagrass. Seagrass also filters pathogens in the waters and clears them. So the seagrass absorbs wave energy and prevents any turbidity that may create murky waters, which is why we have the crystal clear blue waters here in the Bahamas. Finally, the Bahamas acquires 50% of their GDP from the tourism economy, and seagrass contributes to this economic gain because many of the adult organisms that are found on the coastal reefs and are a huge tourist attraction grow up in seagrass and find a home in the seagrass beds.
So here are our objectives and what we hope to answer. Our objectives were to test seagrass around southern Eleuthera, which is a primary producer, and by doing this, we hope to see that there was no human impact affecting the seagrass populations. However, we tested seagrass in close proximity to humans as well as isolated to see if there was a difference. When we initially tested the seagrass for percentage coverage, we hypothesized that there would be a difference in the coverage, as well as when we tested the growth rate, we hypothesized that the growth rate would increase due to the potential nutrients in the water. As we're looking at these objectives, we hope to potentially be able to correlate growth rate and percentage coverage to distance to humans and temperature.
So for our research, we had 3 questions that we were trying to answer. The first one being is, does anthropogenic nutrient loading affect the growth rate and biomass of Thalassia testudinum in creeks in southern Eleuthera? The second is how do abiotic factors affect the growth rate and biomass of Thalassia testudinum in the creeks in southern Eleuthera? And the third one is, what is the difference between the growth rate and biomass of Thalassia testudinum in the different creeks in southern Eleuthera?
So in order for us to do our research, we look at 3 primary locations in southern Eleuthera, and one of which is isolated from human activity, and 2 are adjacent to human activity. And within these 3 different locations, we have 10 different study sites. The first location that we look at is Deep Creek Settlement, where we have 3 different study sites. The first one is Rollins Creek and Deep Creek, which is close to Deep Creek Settlement, and the last one is Plum Creek. Then we have Winding Bay and Half Sound, both of which are separated into north and south, so take up 4 of our study sites, and they are adjacent to Holiday Homes. Then we have Starved Creek and Poison Creek, which are isolated from Rock Sound, and these are 2 other of our study sites. Within these study sites, we have subsites that are generated by an ArcGIS GPS program created by a grid system that makes the subsites each 100 meters apart. Then each of these subsites is in close or far proximity from human activity and ranges from having 11 to 453 homes surrounding it. The subsites are commonly in low-lying coastlines and surrounded by mangroves.
So at the subsites that Olive just mentioned, we lay down a quadrat, we measure the depth of the water, and we take a photograph of the quadrat. Following this, we record the percentage cover of the 3 main types of seagrass in addition to macroalgae. And finally, we record the bottom sediment type. At our 10 main sites, we also installed permanent temperature loggers over the course of our study. And these were important because seagrass tends to thrive in areas of high fluctuation, like the shallow creeks we were just mentioning. So lastly, we're also doing growth rate studies on our project. So how this worked was we chose 3 subsites of each of our 10 main sites, which were chosen based on an already observed high percentage of turtlegrass. So at each of these subsites, we installed a piece of permanent rebar to make them easily accessible to find. And when we returned, we laid down a quadrat, and we poked holes in the sheath, so the bottom of the blade, of a minimum of 15 blades of seagrass, which permanently scarred their tissue, making it visible to analyze growth rate. So 2 weeks later, we returned to these sites, and we collected half of the quadrat. And then 2 weeks later, we returned and collected the rest of the quadrat, which was then used for data analysis.
From the habitat mapping we did, we were able to create a hotspot analysis. And this is the hotspot analysis for Poison Creek on the left and Starved Creek on the right. And here, the red dots are a 99% chance that there's seagrass there. The white are a 50% chance, and blue would be a less than 5% chance. And this is for the turtlegrass. And you can see there's a lot of turtlegrass up far in the creek, and this is most likely due to temperature fluctuations with the tides. And here's one for the macroalgae there, and it's interesting to note, in the creek, it's in a different spot, mostly likely due to competition for space. And from the growth rate, as Claire mentioned, we have this data here, and so in Rock Sound, Deep Creek, and Winding Bay, those are the 3 locations adjacent to human population, and we see they have the largest growth rate over the course of 4 weeks. And then Plum Creek and Half Sound are connected to those creeks, so they also have a high growth rate. And the 2 isolated ones, Poison and Starved, are the lowest growth rate. And here's the graph of those put together with the average from each of them. And with the permanent temperature loggers, we put them out for about a total of 2 months. And there's a lot of fluctuation you see, all the way from 22 up to even 30 sometimes. And this is in Celsius, so that's a large temperature range. And one really cool thing is April 23rd, there was a large hailstorm here in the Bahamas for the first one in about 40 years. And there's a large cold front beforehand, and then the temperature immediately rises back up. And here's just a random day we chose. and it shows the temperature fluctuation within a day. And that's key because seagrass, they love temperature fluctuation and it helps them grow faster.
So for the hotspot analysis, we saw that the seagrass was further up the creek than the algae. And this may be due because of the temperature and water fluctuation, which means that there is constant fresh salt water being flushed through the creek, which allows new nutrients for the seagrass. However, this constant flow of fresh water may prove to be detrimental to the survival of the seagrass because if the nutrients continue and allow the seagrass to continue to grow at the rate it is, and if it is not controlled or grazed enough by green sea turtles, the seagrass will turn to be an unpalatable or woody substance that the green sea turtles cannot eat, thus unbalancing the ecosystem. The way we can see this happening is through growth rate data and nutrient data, and then we can correlate to how long or if this is happening to the seagrass. The significance of our research is the fact that seagrass is a global primary producer. Because of this, we'd like to take our research to a global scale. From our research, we'll be able to hypothesize further studies such as in similar locations such as these creeks across worldwide, how is anthropogenic nutrient loading affecting the seagrass there. And what is interesting about that is if seagrass is not present within that area, the opportunity presents itself to use macroalgae or kelp because they serve a similar purpose as seagrass. And then from this, we hopefully may be able to see a correlation to how and why the seagrass is struggling, thus finding the effector and then hopefully be able to reduce the effector and save a lot of coastal habitats. So we'd like to acknowledge the Island School and CEI, as well as our research advisors, Logan Zeinhardt and Izzy Lake, and the CEI interns that helped us in the field. Here's our works cited, and any questions.
The question was, is there any connection to the names Poison and Starved Creek? Not that I'm aware of. I'm not quite sure why they're named that.
Does the higher nutrient loads, does it cause the seagrass to become fibrous faster, or is it just that it causes more growth that gets grazed less and matures at the normal rate?
So it does a little. Oh, the question was if the nutrients filters through, or like the high nutrients, does that make it a more fibrous seagrass, or if it just grows faster? And so from our research, we found that it does both. So the woody substance is the more fibrous seagrass that is unpalatable for green sea turtles, and then from that, it wouldn't be able to be grazed.
Is that a hypothetical, like you think that would happen, or is that something that you observed, like is it an actual problem here right now?
The question was, is that something that we have observed, or is that an actual problem with the seagrass right now? So we have not observed that, but
Yeah. So there's been research done in other areas showing that the turtles no longer eat the seagrass once it becomes fibrous, and so that's how we know that it's not good for the ecosystem.
How about the temperature block? How do you plant a permanent temperature? What is that?
So the question is, what is a temperature logger and how do you plant it? We actually attached our temperature loggers to a cement block, and we dropped them in each one of our 10 main sites, and then they actually continuously record the temperature of the site.
Trying to find the picture of it. Sorry.
So it's pretty small. Right here?
I just had a quick question in terms of, I know you did a lot of research more kind of on the growth. Did you, during any of the research, do anything about planting new or kind of getting a way to more populate the area with more seagrass, or no?
So the question was, did we do any research on planting new seagrass? And we really did not. What we're focusing on is kind of the more current issue of that it's actually becoming overloaded. So if we can stop things, especially anthropogenic disturbances, so like sewage, runoff, erosion, the seagrass will be healthy, but we didn't need to plant any more.
Did you count how many turtles during your research?
The question was if we counted how many turtles we saw while we were doing research. And we didn't count exactly how many, but every time we saw them, we would mark it down on in a table and to keep track of what, if we saw how many turtles we saw. But it didn't just stated turtles. We also saw a lot of other organisms, which we also marked down and marked where we saw them. Yeah. So the question is, even after the seagrass becomes woody, is it still able to provide those same ecosystem services like photosynthesis and keeping the sediment together? And yes, it is. It's just not as good for the turtles, but it is able to provide those services for the ecosystem. Yeah.
In your research, did you find any correlation between CO2 in the water and seagrass concentrations changing over time?
So the question was, in our research, did we see a correlation between CO2 and the seagrass that we observed?
So we did not observe CO2 directly, but we saw it from nutrients such as sewage, which sewage carries CO2 to the high nutrients. So you could see a correlation through that, but not a direct correlation from CO2 in the water.
And that observation has actually been observed in past studies as well, so it's been done.